All right. Okay, so with data collection, I've got a slide deck, but what I'm going to try to do is go over some of the principles and then just show you a project I'm currently working on now where we're collecting data. And the idea basically here is you want to make sure that you have a good set of rules and instructions to follow. And so that's going to consist of a protocol and a code book and some way of collecting the data and some way of storing the data. These days, if you're running a big study and you try to publish it, a lot of the journals will want to see your protocol because they want to make sure that you're actually doing what you said you were going to do. Now, that seems pretty obvious, but one of the problems that we've had a lot in data analysis is something called publication bias or reporting bias. What that basically means is people carry out a study and then it doesn't work the way they thought it was going to, and then they decide not to publish it. Or they carry out a study and they collect a lot of data on a lot of different outcomes, and some of them come up significant, and so they publish the ones that are significant and not the ones that aren't. Okay, so you can probably think right away what are a couple of problems with that. I do a lot of meta-analysis, which means I'm putting together information from different studies. So let's say there have been 5 studies done on a topic, and 2 of them came up in favor of the new intervention, let's say, and 3 of them didn't. And the 2 that were favorable got published, and the 3 that were unfavorable didn't get published. And so now somebody goes to see what the evidence is, and they find the 2 studies that were published that are both favorable. And so they come out with this big conclusion that both studies that were done were favorable, and they have no idea about the 3 studies that weren't published. Or 5 outcomes are collected and 3 are positive. So let's say those 5 studies looked at the same 5 outcomes, but 2 of the outcomes were significant in each study, but they weren't always the same 2 outcomes. So now you've got this sort of selective publication where you're going to find that each outcome is significant in every study that it was published, but you don't have the non-significant publication. So if you have a protocol where you've said, I'm going to collect data on these 5 outcomes, somebody can now say, all right, well, show me those 5 outcomes. Show me the results of those. But if you don't have the protocol, you don't know what they intended to collect, right? So what is reported is oftentimes not what was collected. So I'm going to show you ways that we set up these protocols and we set up the data collection forms so that these sort of things don't happen, right? And so, for example, I was working on a clinical trial that was published last year, and we had originally said in our protocol we were going to look at 30 different outcomes, I think. And we ended up publishing like 10 of them. And so the reviewers very rightfully asked, well, what about the other 20? And it turned out, well, the other 20 were things that we were going to eventually publish, but they were sort of secondary outcomes and they weren't pertinent to this particular paper. But they still wanted to see the results because they wanted to make sure that we weren't just trying to hide them somehow because they didn't come out the way we thought they would. All right, so some of these slides are a little bit old in the sense that these are not necessarily programs that we use anymore, but the principle holds. So you have data coming from different sources. So one of those sources might be records. So if you're doing clinical research these days, a lot of hospitals are collecting data electronically. So they have electronic health records. You may have seen this when you go to see your doctor. They're typing things in. I was just at the dermatologist, and they're taking pictures of my face because I'm fair-skinned and I get sunburned. And so they want to see if there's any little lesions on the face, and then they can compare it to last year. Well, last year you had it here, this year you have it here, right? So all this stuff, images, data on your past medical history, lab results, all this stuff is stored, and there are different systems, and there are different ways of interacting with those systems. So you might be doing a multi-hospital study where there's data coming from 5 different electronic systems, and they've got to integrate those somehow. So that's one source of data. Another source of data might be case report forms. So I'm going to show you some for the study we're doing, where people actually fill information out and put it into a case report form, and then that gets sent somehow to some centralized source. Then finally, you might have imaging data. You might have so ECGs are electrocardiograms, which are heart tracings, right? So there are 12 different leads that are put on your heart. They give you a tracing. You might have the raw data. You might have some sort of summarized data from that. Or you might have data from a cancer registry. You might have data from there's many other sources of data. So these all are going to get pulled together somehow. And so you're going to put them into some kind of a database. Now, what sometimes people do is they just put them into a spreadsheet, right? You can put them into an Excel spreadsheet. That's pretty straightforward. But again, if you're interfacing with all these different systems, that may be difficult to put into a spreadsheet. So, in the old days, we used Microsoft Access. I think it's still around, although I don't know if anybody uses it anymore. But there's a lot of these systems like SQL and so on. You might be scraping data off the web. So you're going to have to put all this information, and one of the programs that's used a lot these days is called REDCap. Some of you might have heard of it or used it. We have courses, actually, if you're interested in learning, they have courses over at the medical school that you can take through the Clinical Translational Research Program, CTR. If you don't have access to their newsletter, that's actually a good thing to get because there's a lot of good resources through that. So Advanced CTR. I'll send out an announcement where the website is, but they have courses in REDCap and so forth. And I've often invited one of their staff to come over and talk about it. I think I might just do it on my own this year, but it's a good program, and it allows you to set up a database pretty easily and store the information. And a lot of studies these days use it. And then you'd bring all that data into maybe some kind of a file that you'd read into a program like SAS or Stata or R or whatever, and then you'd make a data set, and then you could do the analysis and get results. So that's kind of the process that we go through. It's become more and more important because we have so much data now. And so one of the things we're going to talk about is how we make sure that the data that we bring into these systems is actually collated correctly. So a lot of times what people do if they're dealing with Excel files and they don't really know what they're doing is that they will start to sort and transform things, and they end up mixing up the rows and the columns. For example, they might sort something and then they'll sort something else and then they'll merge it together, and then things get jumbled. So you have to be careful. That's why it's good to have a good database so that you have the source data and you can always go back to it if you need to. Okay, so some of the questions you want to think about when you're doing this is, what are the databases that you might be using? What are their capabilities? How are the data going to be stored? We talked about how it can come from different sources. They can be stored in character form, numeric form, dates. Dealing with dates is always an issue because different systems have different ways of dealing with dates. And you want to make sure that they're consistent. And then, of course, there are different ways that programs that you can use. So we've talked a little bit about making things reproducible, and so it's always good to keep things in as simple a format as possible: text files, CSV files. Most of these good systems like REDCap or SQL and so on, they have ways of exporting data into different formats, and so they will do all that sort of thing for you, but it's always a good idea once it's exported to make sure that it's in the format you think it should be. Now, when the data are coming from different sources, or even sometimes in the same source, for example, in a spreadsheet, you can have different tables in or different worksheets in a file, and you're going to need to merge those together, and you want to link them. And so it's good to have a system that actually promotes such linking. So most relational databases allow you to do that. And there's some kind of a variable, a key that you merge on, right? So typically that key is something like the ID number, and you want to make sure that those ID numbers are related. So if you take the data from an electronic health record and you also have data from case report forms, you typically will want to get the data from the electronic health form that relates to the individuals who are in your study, who are presumably you've done the case report forms on. And so you want to pull those records off, and you want to make sure that the IDs link so that when you link the sources together, they're linked properly. And so that's the key issue, which can oftentimes mess you up because, for example, people might have typed in the wrong number. So you've probably seen this in your personal data, right? When you go to some account and somebody's entered your name in wrong, and you can't link to it because they misspelled your name, or they put the wrong birthday, or you've got like some other identification wrong. So that's important here too, and we'll talk about how you can tie things together on different fields. Now, what are the data that you're storing? You're storing IDs, which are sort of identifiers of people, and then information on those people, which we can think of as variables, which are kind of like the columns in your spreadsheet. And so those variables have different attributes, right? So there's a variable name. Each variable has a certain type. It's either numeric or a character or a date. It's got a certain length. So the database is going to store this in a default fashion, but you can oftentimes fix this up. So for example, do you want it to have, if it's a character field, do you want it to have a certain length? Do you want the numeric value to be a floating integer or a floating point number? Do you want it to have single precision or double precision? Now, if you're getting into real programming, this is going to be important because when you carry data along, so single precision means that it's stored basically with a certain fixed number of binary digits, and if it's double precision, it is double that. And so typically, if you want the number to be accurately stored, double precision is good to have. Now, with most sort of systems that you deal with, like R, you don't have to worry too much about that. But if you start programming at a level like with C or Java or something, you might need to know these attributes of variables. And it's important because if you don't have enough precision, you can lose integers. You can lose significant digits when you're computing, and so numbers might not add up the way you think they should. We won't be getting into that, but if you take a programming course, of course, that's sort of thing that you learn about. The format. So in a spreadsheet, right? If you're working in Excel and you want the data to be displayed as decimals, for example, let's say you're recording a grade point average and you want one decimal place, you can make the column to have one decimal place. That's the format. So you can do the same thing in a program. So R, for example, you can tell it how to display the numbers on the screen. So my kind of rule of thumb is don't display more digits than you need to. Sometimes, for example, the default might be 6 digits of precision or 6 decimal places. And so people will print out these regression coefficients that have 6 decimal places when clearly they're not that accurate. I typically will try to make it as simple as possible. A good rule of thumb is to try to make things integers if you can. So for example, if you're reporting ages, a lot of times papers you'll see the average age was 69.37. Well, it really doesn't matter whether it's 69 or 68, right? It's probably rounding it to an integer makes it easier to read and also easier to kind of understand. So you could usually in these programs go to any kind of level you want, but try to just use numbers that you actually believe. You probably don't believe the 6 decimal place. And then how the data are stored in a database, it doesn't really matter too much as long as you can access them, but it will be important sometimes with how you report them. So sometimes you might want to order variables from largest to smallest. Sometimes you might order them numerically, sometimes by category. And so those sort of things are important to think about. And then finally, the label. The label is what's displayed in the program. So for example, if you're doing a regression and you have an independent variable that's, let's say, systolic blood pressure, you might want to call the variable SBP, so you don't have to type systolic blood pressure every time you use the variable in a model. But SBP may not be something that people outside the field know that well, so you might want to actually have it print out as systolic blood pressure so you can make that a label, and that's good to know to have both the variable name and the label. A lot of times I see homeworks where people have created a variable called age underscore 30, which kind of means like, are you over 30 or under 30? And then they print that out, for their homework. And so we typically will circle that and say, this doesn't make any sense. What does this mean? Sometimes people will footnote it because sometimes tables are too wide to display. And so you do need to abbreviate things, but then you should always footnote them so that people know what they mean. So again, the key or the ID is the thing you're linking on. So how are you going to combine data from more than one table? And typically that's going to be some kind of a linker. It could be the ID number. It could be the ID number and the name. If you're collecting data in a survey and you do stratified sampling, so you're collecting data at a household level where household is nested within, let's say, census block or something like that, you may need several levels of this to be able to uniquely identify an individual. But every record should have a unique key so that you can merge them. And so this particular study that we did years ago, we were combining electrocardiogram data, which was in a spreadsheet, with clinical data that were in a database, and 30-day follow-up clinical data, which were in a different database. And so all those were stored where the individual was identified uniquely by an ID. And so when we merged them, we merged on that. And so therefore, it took all the information from those databases that referred to that individual and put them all together. Now, sometimes the individual will be in one database and not another, so you want to make sure that if they're not in one database, that those particular records come up as missing, right? So, and again, with missing values, these databases could have data stored differently as different missing values. So for example, a lot of times in spreadsheets, when people are entering data, if the data are missing, they will just leave it blank, right? And so that gets read into R in a particular way, or into SAS maybe in a different way. But you want to make sure that you distinguish between what's really missing and what's not available. So some variables just may not be applicable. For example, if one of your variables is the individual pregnant or not, that's not going to apply to males. It will apply to females. So you want to make sure that the missing for females means something different from missing for males. So just be clear on those sort of things to make sure that you are not confusing data that are missing with data that are unavailable. So again, a file will have observations. Each record refers to a line or observation. We talked a little bit about the wide and the narrow format, remember? So the wide format would be, I have one line, which is the person's ID, let's say person 1, and then I have a lot of information on them. For example, it's a longitudinal study, so I might have their outcome Y at baseline, and I might have their outcome Y at 30 days, and I might have their outcome Y at 3 months, and so forth. And so this is all laid out this way, and I might have 10 such variables collected longitudinally, so it's a very wide database with one row per individual. So this is the wide format, right? The narrow format would be, I have an ID and then I have a time. So for person 1 at baseline, I have their different outcomes. And then I have person 1 at, say, 30 days, and now I have their outcomes at 30 days, right? So now I've put one record per time period, and so this is going to be a much narrower database because I only need, instead of needing a column for each time point, I only need one column for each variable at a specific time point. And so this is the narrow format, right? So both of these are different ways of storing things. So in this case, each row is a particular individual. Here it's a particular visit for that individual. I might also have another database. So this might be my individual database. I might have another database which is let me call it institutional or hospital level. So for example, let's say I'm looking at data across different hospitals. Now, this might be information on a particular hospital, like hospital, ABC, and I have things like number of beds that the hospital has, number of, let's say I'm doing an OBGYN study, maybe number of deliveries, baby deliveries that they do, number of nursing on staff, or number of nurses per individual, per patient. And now I might want to merge this data with this data. So I should have a variable over here, which is hospital, right? And now that becomes my ID that I link here. So let's say this is hospital ABC. This is, and then the next person is in hospital BCD. So this is going to link to that line, and then the next person gets linked to this line, and then I tack on this information for that person. So now I can link together the patient level data with the hospital level data, right? So this is a very common type of thing that we do. Or these could be different studies, could be different sites in a multi-center study. They could be different towns if you're doing a study across the country. All right, so when we merge the data now, we can merge the data in a couple of ways. So a one-to-one merge means that every table has one row per subject, and you're basically just merging, you're just taking each line that applies to the same subject. So for example, here we have one record per person. Let's say it's a case report from a database, and then we're merging all that information in that person. In the second case, we have a one-to-many merge. So each table has one row per subject, or one table could have one row per subject, but other tables could have multiple rows per subject. So for example, in the longitudinal database, I've got different times. Now, these times might not even be consistent between databases. I could have one database which has got data every month, and another database which has a different set of variables which are collected every year, for example. And so I need to be careful when I merge them that I'm merging on the same IDs and I'm getting the right variables to do that. And so programs will have, like, R has a merge function, but you just have to be careful when you're doing it to make sure that you're merging with the right characteristics. So for example, here we have one database which has the age and the gender and when they got their electrocardiogram, and then the other database has the actual electrocardiogram information. Like for example, one of the pieces of information you pull off electrocardiogram is what's called the ST segment, which is the amount of time between the S wave and the T wave, and if that wave is elevated, that's indicated that the person might be having a heart attack. So you label that whether or not they have an ST segment elevation, and then LBBB is left branch bundle block. So that's again a particular kind of restriction in your heart, and that's a 0-1 variable as well. So here we want to merge the information so we'll have another database which has their age, their sex, and then this electrocardiogram information. And so here you can see that we are merging we're gonna leave it as multi lines per subject, but we're going to make sure that you'll notice that the age, for example, on patient A2, the age and the gender information is recorded twice because it's the same person, right? And then we have another person who's in one database but not in the other, so we have their electrocardiogram information, but we don't have their age and their sex. And if that sort of thing happens, and you get that and you say, I'm confused because I should have the age and the sex on everybody in the database, then what you might want to do is make sure that this individual was not wrongly coded, right? There could be a wrong ID there. Maybe that should have been A3, or maybe that should have been A4, and there is an A4 in the other database. And so that sort of brings another good rule of thumb is when you're collecting the data, make sure that you're checking it periodically. So the study that I'm going to show you, so we're collecting data on people in Bangladesh. They're coming into a hospital, and certain numbers are being recruited every day. And every month we run a check to make sure that there are no errors in the database. For example, there's no people who are over the age of 100. Or if there are, that we check to make sure that those are valid numbers, or that they don't have a blood pressure that's 300, or that they don't have, it says that they traveled 2,000 miles to get to the hospital or something like that, right? Somebody typed in the wrong number, or they forgot to put a decimal place or something. So you want to check those because it's a lot easier to catch them right after the data entry has been done incorrectly than to try to catch it at the end of the study 2 years later when you've got to go back and you might have questions and then the person's no longer around. Or in particular, if you've got patient-reported outcomes and they're reported incorrectly, it's a lot easier to go back to the individual and say, hey, you said that you ate 3 meals yesterday, but in the database it's recorded as 2, like which is correct? That's a lot easier to check when you've got the data right at hand. So here, for example, we have three different records on the same person. Now, in this case, we may only want one time, and we might want to make sure that these 2 times match. Well, in this case, they don't, right? This information from this database, which has the ECG information, and this information, which has the age and the sex, and they've got 2 times recorded, which are different. They should match, but they don't. Notice they match here, but they don't match here. And that may be because they were either entered incorrectly, or maybe this person actually had 2 electrocardiograms taken. And so now the question is, well, do you want the first one or do you want the second one? And so you want to make sure that these things match up. And it's a good idea, again, to check those right away if you can. So what we try to do is we try to code data entry systems these days that are electronic, but that also have built-in checks. So for example, you either have a pull-down menu. So for example, you only can, if you have sex, and you have to decide how you want to code that, male, female, other. There's a variety of ways that we do that now. So you want to make sure that you've got that right up front, because what you don't want to have happen is somebody to type in something and they type it in incorrectly. Or what happened to me, one of the very first jobs I had, I was asked to clean a database, and I realized that a variable had been coded in 15 different ways because the number the word yes had been written in all caps, in all smalls, cap Y, small es, cap Y alone, and a computer doesn't know that those are different. So when you do a frequency table, you find that there are 15 different ways of coding yes. So you can get through that by just making sure that your program only allows yes or no. And if there's a different answer, it either returns an error or just doesn't, or prompts the person to re-enter it. Okay, the next thing is documentation. So now you've got this database set up. You need to give the data to your analyst. You want to make sure the analyst knows what it is. So you want to have what's called a code book, which is just a set of instructions which tells you what every variable is, what it's named, and what the possible responses are. And so you want to make sure these are documented. It has what the variable names are, what the attributes of the variable are, what the possible responses are, and then what to do in case there's a mistake. So you can have a lot of different types of documentation. I'll show you a couple of examples. It could be a case report form. It could be a code book. It could be a documentation. For example, in SAS, there's a procedure called contents, which will print out all the variable names and all the variable types and so on. In R, you can do a dim name statement to get a list of the variable names, and then you can get the attributes of those variables as well. And sometimes you might actually want to include summaries of the data in the documentation, like what are the different possible responses, and what does it look like that people have actually responded. So here's a case report form from an old study. Again, I'll show you some of our case report forms. So this is not important to know the details on this. But notice here that you've got the variable names on the side. So, for example, the ID number gets written or typed in, and that's stored in a variable called PATID. And then here in the gender, in this case, they only allowed male and female, as this was an old study, and so it's coded in the variable gender. Age is given three places, so it's an integer, and that's stored, and so forth. You'll notice then, how did the patient get to the hospital? And they give you 8 choices here, and then they give you 8 is other, and you can type in other. But the idea here is to try to get the most obvious ways of getting to the hospital. You know, did you drive by yourself? Were you driven by somebody else? Did you take a taxi, public transportation? And code those so that you don't have lots and lots of different entries that you then have to clean up later. And then this would be, for example, in SAS, how a contents would be labeled. You'd have the variable name, the type, the length, the format, and the label. Okay, so the coding, we want to make sure the data are accurate and equally analyzed. We want to make the codes easy for somebody entering the data on a keyboard or whatever. And so if you are going to ask people to enter a code, try to make sure that it's easy. So, for example, if something is, what's the level of your stress? Is it low, medium, or high? It's easier to do L, M, and H, or versus something like ABC, which is just very generic, right? What you want is you want the person to be able to look at the database and know from the variable name what the variable is, and also from the entries what the response is. So if you're coding gender as 1 and 2, that's not very helpful, because then what's 1 and what's 2? If you code it as M and F, it's a little bit easier. If you code it M, F, and T, for example, for transgender, then that's even more obvious to people what that means. But again, you want to have a code book then, so that people know, because somebody might have coded, G, and like, what does G mean? And for somebody, it might mean gay, but somebody might have put in G and like, no, I don't want that. I don't want the letter G. I want something else. But you got to tell them what that is, right? So that you don't get these errors later on that you need to clean up, and then all kinds of missing data thrown in. Okay, so the coding. All right, so as I said, pull down and check boxes are helpful. You're going to find that you want, in the end, your database to have numbers, zeros and ones. It's a lot easier to code with zeros and ones when you're in R. But you might not want the data saved that way, because they might not be able to read. So you got to make sure that if you're coding something like as yes and no, y and n, and you want the y to be 1 and the n to be no, just make sure that that's in your code somewhere, so that calculation is made automatically and somebody doesn't have to do it. I've mentioned the different ways of coding things, which can be problematic. Code as you go along. You may need to make changes to your code book as well, as you find entries that you hadn't thought about that come in. If you're doing hospital type data, some of you may be aware that hospitals code conditions with things called ICD numbers. The current standard is ICD-10. So, for example, if you have appendicitis, there's like a code for that, right? And so that's used both for billing purposes and diagnostic purposes. So it's good to use standard codes like that. Don't try to make your own up. Beware of character versus numeric fields. We talked about that last class, because you can't add characters, but you can add numeric fields. Understand how your software handles missing data. We talked again about that last class, but also how it's coded. Is NA what it's using, or is it a blank, or is it a 999? A lot of different ways of doing that, and make sure that that's done consistently. And then missing value indicators. Sometimes people like to have a variable like, is it missing or not? That could be useful when you're doing missing data analysis. Sometimes people just like to use those indicators in their models. That can be useful, but it can also there might be better ways of doing it. Okay, so for example, if you've got a racial and ethnicity category here, be efficient. Use binary logic. So this might be the way you want people to enter it as a checkbox, and then you have to decide when you're coding, how you're going to code that. So for example, the racial category here has 5 different categories. If you're going to treat that as a categorical variable in an analysis, you may want to have indicator variables for each possible combination so that you could easily do the analysis. You might also want to have a separate one for ethnicity. You then might want to combine them. For example, you might want to have white Hispanic, black Hispanic, and so forth. And so you need to think about that as maybe you don't need it in the coding form, but you will need it in the analytic form. So here's an example where they actually coded things in not using binary logic, but actually had it as a sort of a categorical variable with lots of different levels. That can be useful for the purposes of collecting the data, but it may not be as useful for analyzing the data. And it makes the form much more complicated because now people have to fill out lots of different combinations. Dates and times, be careful of these because they're stored as a number in most databases. So if you actually are using the date fields and they're coded that way, they'll appear as dates. But if you don't tell it it's a date field and you start adding dates together or subtracting dates to see what the difference is, then these things automatically get converted to numbers. And different systems will use different bases. So for example, in SAS, day 0 is January 1, 1960, but in Excel it's January 1, 1900. And so therefore, January 1, 1960 is actually day 21,916, and R actually uses January 1, 1970. So again, if you are transferring numbers, if you're combining numbers from different databases, like you have a SAS database that you want to combine with an R database, just be careful on dates because you might have the same date that's stored 2 different ways in different databases, and then when you combine them, it may get mixed up as to which one it's talking about. So for example, if you're dealing with day 1 in SAS, that's in 1960, and if you're combining that with a day 1 in R, that's 1970, and so when you put them together, you may not know which date it's talking about, or it merges date 1 and it converts it from 1960 to 1970 or something like that. So again, there's ways of dealing with dates in these programs, but just be careful. You can, of course, display it in various ways, and storing it as a number can be convenient for the program, but again, the display is not necessarily the same way that it's stored internally. All right, so I'm going to skip through these because these are just different case report forms that I had for another database that used genetic information combined with clinical information, combined with laboratory information. And so we had all these different forms that we used for different enrollment purposes, and then we had to merge these together into a database. So this just shows you, like, for different studies, these can be quite complicated. But again, I'm going to give you an example from one I'm working on right now. Okay, then the next step is cleaning the data, right? So you've hopefully avoided most of the problems with the way you've collected the data and you've coded it and you've documented it. But mistakes do come in, particularly if you're getting data from other people who might not have been as careful as you were. So years ago, I did an analysis, meta-analysis actually, where we were combining data from 11 clinical trials, and they were all over the world. And we originally asked them for their data, and we sent them out a form that we wanted them to put their data in. Like, we wanted these particular variables in these particular forms. So what happened was a couple of the teams actually sent it to us in that form. A bunch of other teams said, well, actually, we've got this SAS database, so we'll just give you our SAS database. And then other teams had text files or paper forms. This was a long time ago. And so it kind of became a nightmare because every group was sending us data in different format. We finally realized that it was probably just easiest to use the databases because they had all the information and we could clean them. And so then we did a lot of checking because there were a lot of values that were wrong in the databases. these might have been variables that were never used in the analyses, or they may have been things that they overlooked that they missed. And so we had to do a lot of cleaning, and it took a long time. And it was actually interesting because we applied for funding for this project, and the first time we applied for funding, we applied for money to clean the data. And I actually remember on one of the grant replies, the comments back to us, they said, well, this budget item is not necessary because they're getting completed databases for people, so all they have to do is merge them together. Why do they need any money to merge the databases together? They were just thinking it was a simple like computer statement, right? And what they didn't realize was that we were getting a lot of medication information, and so that was all often in text fields. You might say, for example, 3x/d, which means 3 times per day, or it might be QID, which means 4 times a day, or BID, twice a day. So that's actually you need some sort of text mining, right? We didn't have text mining tools back then, so we had to actually go through and code that. So we had to create a variable which is number of pills per day, number of times per day, number of what was the dosage, and then calculate that. And then I remember one of the studies had been done in Sweden, and this all came across in Swedish. So we had to figure out how to translate all that stuff. And so this can be really pretty expensive to do, and you need expertise. Okay, so I said how to avoid these things. Well, if it's your own study, you want to make sure that you're checking it as you're collecting the data, checking your case report forms prior to data entry. Build in things like range check. So range check says if you're entering an age, if it's outside the range of 0 to, let's say, 110, don't allow it. Or if a logical check might say, if the person enters that they're male, then don't show them the following questions because they're not applicable and you don't want to have data entry errors. And make your data entry easy. Just, check boxes, simple little, if it's an age, maybe just give them 3 spaces to fill it in or something like that. And then double data entry is always a good idea too. That means you want to have, at least in some of your entry, you want to have 2 people entering it so you can make sure that it's being entered correctly. That can be very helpful at the beginning of a study because you might have a new research assistant who's never done this before or hasn't worked in this area, and they make some sort of logical mistake, which might be a pretty common one to make. They're misinterpreting a question or something, and so you can catch that easy and teach them how to do it correctly. And then other things you can do are, for example, you can make plots. So you could make, a histogram or a bar chart or something just to make sure the data look right, particularly if you're collecting a lot of data. How many values are missing for each variable, and various, as I say, various kinds of logical checks. So this was the meta-analysis I was talking about that we combined 11 studies from. And so we had some research questions that we wanted to ask by combining these data. We wanted to know, for example, if this was a comparison of ACE inhibitors, which is a kind of blood pressure lowering medication, versus non-ACE inhibitors, placebo, for example. We wanted to know whether the effect was in these individuals who had kidney disease was basically the reason it was working was because it lowered their blood pressure and the protein in their urine, or was it actually, did it do that, but it also had sort of an additional effect? We wanted to know if it worked equally well for all patients, or was it better in certain subgroups, and what was the optimal dosing, and various other things. And so this is a, hopefully you can see that, a spreadsheet of what the different databases, as I said, came in. So the first database, which was a study of 121 people, we got it in a program called DBase. The data were entered in Italian. There were 1,663 visits among these 121 people. They used a particular ACE inhibitor called captopril. The comparison was to a calcium channel blocker. They recorded data. So the average age of people in that database was 55. 61% of the individuals were male. There was 3 years of follow-up and various other information. You can see that we got them in various different formats in various different languages with different sample sizes. Some of them had a lot of follow-up data, some of them only had a little bit of follow-up data, and they were using different drugs. This is another, so this is again in one of the papers that I put into the syllabus. So again, similar information. Now the studies are arrayed as columns, and you can see, for example, some of the studies were blinded, some were not, and gives you the different doses and so forth. So these were this is all information that was sort of different across these studies and enabled us to do some additional analyses that the studies alone couldn't do. So when we put it all together, we ended up with 4 data tables. One was a patient table, which basically just provided information on each person. Another was a study table. Another was an outcomes table. What were the different outcomes? Another one was a visits table, like information on every time the person visited the doctor. So there were 31,000 records there. There was another medications table, which says what medications were they on and when did they change them. And so we put all that sort of stuff together into a database, and that database had like 5 different types of information, and then we had to merge that all together. So we had to. So some of the complicating factors that arose here is that. So let's say you have a study, right, and you have the paper that the people published, and then they give you their database. So you want to make sure that you can reproduce the information that's in the study. That's the first step. So let's say that the study says that there were 155 people in the study. And let's say 80 of them were males and 75 were females. And then you get the database, and the database has 157 records, of whom 84 are females and 67 are males, if I did that calculation correctly. I don't think I did. But anyhow, it doesn't match up. Like, the total numbers don't match up and the subgroups don't match up. So now what do you do? Well, clearly there's a mistake somewhere. So that was one of the things we had to do. We had to go back in and see when we couldn't match up from the original paper, like what was going on. So I remember there was one paper in particular where there were 260 people in the database, but 257 in the publication. It turned out that they'd excluded 3 people for some reason, but they never said that in the paper. So we only found out about it because we had their data. And I don't know why they excluded 3 people. I mean, they might have actually been reasonable exclusions. Maybe they didn't satisfy their inclusion criteria and they got into this study incorrectly or whatever. But it could have also been an error on their part, right, that they should have analyzed those individuals and they didn't. So one of the things that you'll hear about is what's called an intent to treat analysis. So an intent to treat analysis means that I'm going to analyze everybody in the group to which they were originally assigned. So in a randomized trial, you randomize people to 2 groups, and you analyze everybody as the way they were randomized, even if they didn't necessarily do what they were supposed to do. So if it's a medication trial, and they're supposed to take a drug in one group and not take the drug in the other group, sometimes people in the non-drug group will go and get the drug somewhere else, and they'll start taking it, or people in the drug group won't take it. But you should always, intent to treat means you analyze them the way they were randomized. A protocol analysis says analyze them the way the protocol said to do it. So if the protocol says that you're supposed to take this medication every day, then you only analyze people in the treatment group if they actually took it according to the protocol. And then there's other ways of analyzing the data too. And so these can all lead to different analyses, and so you have to make sure that that you do it correctly in the analysis, but also when you read the paper that you know how they did it. And a lot of times it's done incorrectly. So for example, you will have a flow chart which says we recruited 100 people, of whom we randomized 90, of whom we analyzed 80. Right? And they didn't analyze 10 people because these people dropped out of the study, but they actually were randomized. And so they're really missing data, and then you have to figure out what's the right way to do it. So why can it be a mistake to analyze the data different way from the way it was intended? Well, let's say you randomize 90 people, and there's 45 in each of your treatment groups, and 10 of the people in the treatment group withdraw because they have an adverse reaction to the medication. So your final analysis has 45 in the control group and 35 in the treated group because you ignore the 10 that dropped out. And let's say the treated individuals do really, really well. So what's the conclusion of your study going to be? It's going to be that this treatment worked, right? However, 10 of the 45 people you originally randomized dropped out, which means that they couldn't have done very well in that study, right? So maybe you impute no benefit to those people, and now maybe that difference that you've observed goes away completely, right? So if you publish the data with just the people who completed, you might actually give the wrong answer in some sense. Or maybe the right answer is, if you can tolerate this drug, take it, but if you can't, then do something else. And maybe that's the right message, rather than everybody should take this drug. Okay, another problem that you run into is that you find missing values or implausible values when you start checking the data. So then you, a lot of times, want to go back to the original data sources and try to figure out what's going on and try to fill in the missing information. You may or may not be able to do that. What we found was a lot of these older studies, the analytic teams no longer existed, right? So let's say you're a company that's running a study and the study ends and you publish it, and now the individuals on the study move on to new teams, or they move to new companies, or they retire or something, and now 5 years later somebody comes back to you and says, can you give me your database? And you say, sure. And now they say, well, you know, this person X is coded here as being 137 years old. I think this is wrong. Can you tell me, should it be 37 or should it be 13? Or should it be 13.7? And usually what happens is the principal investigator will say, I have no idea, because the people who collected the data aren't around anymore and I don't have the original forms, and so that's really hard to find. So another issue we had was reconciling dates. So the dates in some studies are recorded as exact dates. Sometimes they're recorded as a visit date. Sometimes they're recorded as just a month or just a year. Another problem you run into is if you are trying to record dates as the beginning of a study and then time to the next visit. Sometimes the begin date is the date when the person was recruited. Sometimes it's the date when they were enrolled into the study. Sometimes it's the date when they were randomized to the intervention. Sometimes it's the date when they first saw the study team. So again, those have to all be reconciled sometimes. And so in this particular case, 4 of the studies used the medication date as the visit, 5 used a trial defined date, and 2 gave only the month number. So you know, we had to do a little bit of work to get those right. And so sometimes you have to try to change your rules a little bit. Like you want to be very strict, but you can't, because there just isn't enough information there. And so again, this is one of the papers that this is the paper on constructing a clinical trial database that I wrote years ago. And you can read that if you want some information on all the different things that we had to do there to make these data come out right. Right. So a lot of detail here, which I don't think is crucial, because I do want to get to the other example. But just to say that there can be a lot of complications here because of the way different studies define things, the way people record things. Again, it's really important that everybody who's entering data is trained so that they're all doing it in the same way. You know, in this particular case, some of the studies, we asked for follow-up data, but we asked it at particular times, and then because some studies sent us what we'd asked for and other studies just sent us all their data, things didn't always match up. So some studies, for example, might collect data every 2 months, some might collect it every 3 months, some might collect it every 4 months, every 6 months. And so when you do your longitudinal analysis now, the dates aren't going to necessarily match up. Or you might have missing values, like somebody missed their 3-month visit but came in for their 6-month visit, and then you'll have to figure out how you're going to handle that. Another problem is different ways data are collected. So for example, this is a study of kidney function And so one of the kidney variables that's very commonly used is called the GFR, which stands for the glomerular filtration rate, which is the rate at which the kidney filters things out of the blood. And there are different ways of measuring that using different biomarkers and different tests. And those give slightly different numbers or numbers on different scales. And so that's, again, a complication that we had to face here was how do we reconcile these different numbers? And sometimes people have done studies that will relate things together. Or blood pressure can be measured while you're sitting, while you're lying down, or while you're standing, and you'll get a different value for those. Urine protein is, again, measured is an assayed value, and you can assay it either just using sort of what's called a dipstick, which basically just measures kind of whether it's present or absent, but then there's more accurate measures, and they can be in different units and so forth. So those are all things that you have to think about. I talked about the medications and how the language issue was a problem, but we also had problems with dates. So if you want to know, somebody keeps switching medications. Let's say it says that they started a medication on February 10th, and then the next entry in the database is on July 9th they were taking some other medication. So does that mean that they switched at that point, or they had stopped taking the first medication, now they started taking a second medication? So all those kinds of things you have to sort of think about while you're doing this. And then there's a lot of different drugs that are in the same class. So for example, there's 10 different ACE inhibitors. Do you consider them all as the same drug, or are they 10 different drugs? And then how do you handle doses? So if somebody's taking 30 milligrams twice a day, is that the same as 60 milligrams once a day? Or is it a different type of dosage? So is it total dosage, or is it number of times you're taking, and so forth? And then different types of drugs will have, even if they're in the same class, will have different equivalencies. So for example, the clinicians can tell you that 1 milligram of enalapril is equivalent to 1 milligram of benazepril, but it's equivalent to 4 milligrams of cilazapril. So different drugs are given in different concentrations. All right, so that's sort of just a quick kind of thinking about how you collect data and some of the things you need to think about. It's not a simple process, and what's actually interesting as a statistician is that I work a lot with doctors and health experts, and at least in the old days, they focus mostly on collecting the data. The idea is once the data are collected, you guys can just push a button and give me an answer. And of course, we sort of do it the other way. We say, well, the data, once we have a data set, then our work starts. But you really have to think, it's really a big issue in both ways, right? Collecting the data is really, really important. You got to do it right. You got to make sure that you have good collection procedures and you've got a good study design and so forth. But the whole purpose of that is so that you can analyze the data well. And we often say that we can't really fix bad data. Statisticians come up with a lot of different techniques for dealing with bad data, but while we can handle missing data with different types of algorithms, the best way to handle missing data is not to have them in the first place, right? So collecting the data is really, really key. Okay, questions, and I want to show you some of the data that we've got. So let me show you. So this is this Bangladesh study I was talking about. So we call it Narudak, which is a Bangladeshi word, and I'm forgetting now what it means. But anyhow, that's where that comes from. And so the idea here was we wanted to develop. So the idea is people come into these hospitals and they are dehydrated. They've got cholera or they've got some other really horrible disease, and so they've had diarrhea. They've lost a lot of fluid, and so they present to the hospital, and oftentimes in like monsoon season, they're presenting there's a lot of people coming in, right? So you've got to triage these people very quickly. Now, you could basically do one of three things. You look at them and you say, you are really, really sick. You need to go into the hospital immediately, be intubated or whatever. So that's admission. Another one is you can say, you're sick, but you can probably go home. I'm going to give you some medication, or I'm going to give you an oral rehydration solution or something that won't require hospitalization. And then the third thing you can do is say, you know what, you look like you're recovering, you can go home. So think of those as our three main decisions. And the way we're going to determine that is basically by how much they're dehydrated. Now, the problem is that we really don't know how much they're dehydrated until they're actually rehydrated. So if you're losing fluid, it's very hard to tell how much fluid you've lost at that point in time. Once you've been put on treatment and you've regained your weight, we now know how much fluid you lost. So we are collecting data on people who are coming in dehydrated, and then eventually they're going to be rehydrated. We'll measure their weight again. We take the difference, and that's how much they were dehydrated. And then we can classify them into one of these three classes. So we need good data on the weights, both at admission and at follow-up. But from the point of view of treatment, we want to be able to identify those individuals and how much they're dehydrated. So our data are going to tell how much they actually were dehydrated, even though we don't know them at the time. And now the clinicians are going to need to make a triage decision. So how are they going to do that? Well, a lot of times it's just going to be by looking at the individual. For example, what do their eyes look like? Are they sunken? Are their eyes sunken? Is their skin dry? Can they cry? Can they drink fluids? Right. So the worse off you are, the less of this sort of thing you can do. And so the purpose of this study was to create a predictive model that could take such clinical information together with maybe things like how much diarrhea have they had or how long has this been going on, and combine that information together to predict how dehydrated they are. Right? So we're going to be collecting information on how dehydrated they are, collecting this predictive information, and then developing a model. All right, so we're going to develop a model for 3 different age groups. We've already developed one for kids under 5. We're going to develop one for kids who are 5 to 20, and then for adults 20 to 60, and then for people over age of 60, because the idea is that these people may present in different ways. All right, so this is the protocol that was developed. So it was originally a grant proposal. There's now a protocol. So I just want to show you some of the things that we cover in this protocol. So here's the table of contents. And so let's go through what this thing, and there's lots of appendices. Okay, so we have the study information, what the title is, where it's being conducted. We have institutional review boards both in Bangladesh and in Providence. This is the grant number. This is the study staff, the research assistants, the research nurses, and so forth. Then we have a bunch of abbreviations. Okay, then there's a background. So acute diarrhea remains one of the most common and deadly conditions in children and adults worldwide, blah, blah, blah. What are the different ways these are being treated? Why basically is the study being done? All right, now we have the limitations of the research to date, that there have been certain predictive tools, but they don't necessarily work well, or they're hard to administer, or they've been developed in one part of the world, but they don't really work in another part of the world. Then we have the rationale for the study, the idea that we want to develop some kind of a mobile health tool that people could use on the spot, something that will be able to be done in third world countries, so you don't need immediate access to the internet or something like that. It's got to be something that can work quickly. Then we have a summary, then manual. Okay, and then we have the protocol. So the protocol, first of all, has the design. So this is going to be a prospective cohort of adults and children over 5 years of age with acute diarrhea presenting to the rehydration unit of ICDDRB in Dhaka, Bangladesh. And so basically the research staff are going to identify eligible patients on arrival, try to get informed consent from them. The people will be weighed, assessed for clinical signs and symptoms. They will then be rehydrated according to standard protocols, and their weight will be collected and other information will be collected. The hypothesis is that we can develop these predictive diagnostic models and then the study objectives. Then we have our 3 objectives: to employ machine learning techniques to derive and internally validate separate age-specific clinical diagnostic models for assessing dehydration severity in older children and adults and elderly patients, assess the calibration and discrimination of each of our models for prediction, and perform secondary analysis to determine whether differences in the performance of the models based on etiology, nutritional status, or sex as a biological variable. Then we have a timeline, so how we're going to collect people and over what period of time. It's a 5-year study. Now we have our flow chart as to what's going to happen in the workflow. This is how they're going to be screened. So these people are going to present. We're not going to take every single patient because that would be way too many. There's a procedure for randomly selecting people for the study. Then there's the process of how you do the intake of the individuals. And again, this is posted up on the up on Canvas if you want to read it. This is so we're going to use marbles, and they'll be weighted in a certain way so you can tell how many you take. For example, it's turned out that it's hard to get elderly individuals into the study, so we're kind of taking more of the elderly individuals than the younger people. And then these are the inclusion criteria. So they have to be at least 5 years old. They have to present with diarrhea. But if any one of these other things is met, then we exclude them. Then there's an informed consent procedure. Yeah.
Just to clarify with abbreviation, so you have it labeled as SOP, but is this the clinical protocol, not the?
It's kind of both. It's the operating procedures for how we select people into the study, the procedure for how we collect the data, and then how we're actually going to analyze the data and report it. So it's kind of got everything in there. Then there's permissions for kids from the parents, how to obtain the informed consent. So all this goes through the IRB, right? They have to approve all this, and so they want to make sure that people are not being coerced into the study, that they're fully informed of what they're doing and so forth. Then there's a quality control to make sure that it's being done correctly. Okay, then we have the baseline data collection. So immediately after informed consent, the research nurse will transfer the patient to the study scale, weigh the patient to the nearest tenth of a kilogram. If they received IV fluid before their baseline weight, they will record the amount of fluid received and so forth. Then they record the date and the time, their ID number, their full name. Then they ask them a few questions like how long have they had symptoms and how many episodes they've had. Then there's a clinical exam. And so at the clinical exam, they get the time, they get their heart rate, blood pressure, and so forth, their temperature, and there's a whole procedure for how to do this so that it's done consistently. Then they do the clinical exam, they do a skin pinch test, they sit the patient upright, obtain the heart rate, blood pressure, then they evaluate the mental status, then they evaluate the patient's thirst by pouring a small amount of water in a cup and offer it to the patient. Circle either normal or drinks eagerly, depending on how the patient drinks, or refuses, unable to drink if they refuse. Then you evaluate their eyes, then you evaluate the mucous membranes, the radial pulse, the capillary refill, and so forth. And then you ask them a bunch of questions about their gastrointestinal history, how they've been doing, how people in their family have been doing, and so forth. Then there's demographic information that's collected. Then they do some microbiological tests with the blood and the saliva and so forth in the stool sample. And then there's a follow-up where they come back in at specific times. The nurses will check on them every 4 hours. We'll weigh them, make sure that they're getting their fluids and so forth. And then the discharge, right? So that's all carefully, and then they return if necessary. So some of these people are discharged before they're fully rehydrated, so we want them to come back so we can reweigh them. Okay, then we have the data analysis section. So this is the part that I worked on. And so we're talking about how we're actually going to develop these models, some of the ways that we're going to standardize the data files. We're going to take the 2 highest consecutive weight measurements, how we're going to define the dehydration, how we're going to do exploratory analyses, how we're going to derive the models, all the different variables we're going to check, how those variables are coded, and then how we're going to analyze the data. We're going to use regressions with logarithmic transformations, categorical variables, lasso, cart, forward, stepwise. We'll be going through over a lot of these methods, later in the class. And then there's a whole algorithm for how we're going to select the variables, and then how we're going to develop the final models, how we're going to assess whether the models fit well, how we're going to deal with missing data, what we're going to do if we have issues that come up. For example, what happens if we don't have enough people in a certain category or we don't have enough certain outcomes. Then there's the recruitment and how we're going to recruit people, how many people we need for the study. And then there's a whole thing on data management, how the forms are collected and all that kind of stuff. Data entry, data backup, confidentiality, retention of records. Then there's good clinical practices. For example, what does the coordinator have to do? What does the physician have to do? What does the nurse have to do? And various flowcharts, what do the health workers have to do? Then there's protection of human subjects, potential risk, potential benefits, knowledge to be gained, adverse events, what we're going to do if people have a heart attack in the middle of the study, how we're going to report that, how we're going to follow it up, then how we're going to train everybody to collect this information, how we're going to monitor it, data audits that the hospital or the NIH might do, oversight. We have a data safety monitoring committee that will check to make sure that everything's safe and running well. Regulatory issues that the NIH requires, making sure that we have enough women and minorities, and then various appendices with all these forms. Okay, so that is pretty thorough. Now, so here's what the raw data workbook looks like. Well, let me first show you the code book. So here's our code book. We have all these different forms. So for example, form 1 we record the ID, the admission date, the time. So notice I've got the variable name, and then I have the type of variable it is, and then the description. So this is the admission date, which is a date. This is the admission time in hour month format as a time. Here's the patient ID, patient's personal identification number at the hospital as opposed to their study ID, their last name, their first name, their sex coded as male or female, their age as an integer, the hours of diarrhea, duration of diarrhea in hours as an integer, duration of diarrhea in days of over 24 hours, number of episodes in the past 24 hours, their weight, and so forth. And then if we look at the data, actually, where's... let's see, where do I have let me see if I can find the form here. Let's see. Not sure this is the right one. Let's see. No, that's not what I want. There's actually a case report form here. So you can see we have all these different forms. Okay, here's the case report form. All right, so there's like 10 different forms. So here's the first form, which corresponds to the data that were in that code book. Right, so here's the admission date. So notice it's day, month, year, and so they give you two spaces for the day and the month and 4 for the year. The admission time, hour and minutes in 24-hour time, the ID number, the name, the first name, the gender, male or female, age and years. I guess we're not assuming anybody's going to be 100. Number of hours of diarrhea, one. And so if you look over at the database, so here's person 1. They were admitted on March 27 at 7:30. They're a male. Their age is 12 and so forth. And then we have all this other information stored. this goes on and on and on, I think over to like letter I or something. And those correspond to all the different information, all these different forms. So case report form 2 is the clinical exam, the nurse who provides the exam, when the exam was given, and then this is all information from the exam. What's their temperature? What's their respiration rate? What's their heart rate? What's their blood pressure? What's their mental status? These are all the clinical variables that we're interested in collecting data on. So the idea is if they are refusing drink and their skin returns to normal very slowly, and they have sunken eyes, and their mucous membranes are dry, and these are all indications that they may be severely dehydrated. Yeah.
Was this translated into the native language?
Yeah, that is a good question. I think here it's being filled out by the nurses, so the nurses all speak English. So, but normally, yeah, you would want to, if you want the person who's the patient to be responding, you'd have to translate this, right?
So all the medical professionals were assumed to have spoken English?
Yeah, I guess in this case the whole team speaks English. So yeah. But you're right. He would have to translate that otherwise. And so then they do a second exam, and then they collect additional information, for example, on the stool, and so forth. And then they collect background data on the person. So what's their mobile phone number? Are they pregnant or not? Now, stuff about transport time and police station, the reason we're collecting this is we want to know where they live, because one of the issues that came up in this study is we need to know their weight at the end. If they don't come back to get weighed, we want to know why those data are missing so that we can do the appropriate missing data calculation. And so if we don't think the data are missing at random, or even if we do, we need to have information that will allow us to impute. So for example, we're collecting information on how far away they live and what kind of transport they have, because we thought that, well, people who don't have a car, or people who live far away, or people who, for example, if a woman who has 5 children becomes dehydrated, she comes into the hospital, she may not be able to come in back again, right, because she's taking care of her kids. So we want to know that information so that we can appropriately impute the information later if we need to. So we're collecting information on that. And then there's follow-up date, like how much fluid they have, what's their weight at various times, and then what's their weight at discharge, and what was their diagnosis, and then when did they return, did they have difficulties returning, and then what's their final laboratory status. All right, so this is like a big study, but we have the forms, we have the code book, we have the procedure, the protocol, and hopefully everything's thought of. But of course, things will always come up. So again, this is a large study. It's got a lot of support for it. It's an NIH-funded study. But these are the sort of things you should think about. And as an analyst, you may not be doing this collection, but you are going to be giving feedback to how it should be done. So my role in this was to make sure that we got all the information we needed, and that it was in a format that would be easy for us to analyze. Okay, any questions on that? Okay. All right, so I think the office hours should be set up, and we will continue on with data on Wednesday.